So we are almost to the end of chapter 17, but what we would talk about in chapter 17.5 is what happens when something goes wrong here, when there's a mistake and we get a different kind of protein than we intended on from a mutation. So a mutation is a change in the genetic material of a cell or of a virus. It can be based on a lot of different things. Some mutations have little to no effect on the cell and on the organism, and some mutations are very, very devastating and very challenging to the organism. So point mutations are mutations where one base pair is mistaken. One base pair is wrong, and that's all, just 1 point. But one simple change can lead to an entire change in the whole protein and can lead to an abnormal protein which can either still do its job but not as well, or cannot do its job at all. So let's talk about proteins. So sickle cell is an example of a point mutation. Basically, here's the wild type hemoglobin, which means without a mutation, and the template strand is CTT, the non-template strand is GAA, so our mRNA is GAA, and we get glutamic acid for the amino acid. But in the sickle cell patient, sickle cell individual, this T has been substituted for an A, so this base pair is now AT instead of TA. So our mRNA gives us GUA, and our sickle cell hemoglobin has a valine where it used to have a glutamic acid. And so this is significant because the valine causes the hemoglobin to fold differently. Remember, our tertiary and quaternary structure and secondary structure causes the hemoglobin to fold differently, and now it can't do its job anymore. It's a different shape, which causes the entire blood cell to sickle and cause all of the symptoms. And so we can put our point mutations into two general categories. So they will either be nucleotide pair substitutions, which is what we just looked at, we just substituted in a different one, or they can be one or more nucleotide insertions or deletions, which means we add in a nucleotide or we take one out. So a nucleotide pair substitution, like I said, is when we just have replaced this one pair with a different one. And they can be silent, meaning that they do not have any effect on the protein whatsoever. You would never even know that they're there by looking at the protein because of the redundancy in the genetic code, which we'll talk about in a minute. Or they could be missense, meaning it still makes sense, but it's a different sense than it's supposed to make. So those are missense mutations, and they still code for an amino acid. We're still going to keep making the protein, but it's going to be a different amino acid. And these can be really, really devastating, or they can make no difference either. And then nonsense mutations happen when the point mutation, the base pair substitution, suddenly introduces a stop codon that wasn't there before. So halfway through the protein, there's a stop codon. The protein isn't being made anymore, and now the protein is only half made. It's not going to be able to do its job. This almost always leads to a non-functional protein. So nonsense mutations, the protein is nowhere near what it should be. So here are all the examples of the different mutations. So here's the wild type. Like I said, this is the non-mutation. Here's a silent mutation. So right here we have a G, but in the mutation we have an A. So what that gives us is now our mRNA has a U instead of a C, but up here our mRNA was GGC. Here it's GGU. Up here we had glycine, maybe. And down here, we also have glycine because of the redundancy in the genetic code. Remember that there's several different codons usually that code for each amino acid. So if our mutation gives us a codon that also gives us the same amino acid, it's not going to make any difference in the protein. Like, our protein is the same. Met, lyse, phenylalanine, glycine. Same thing down here. Same amino acids, we're going to get the same exact protein. No big deal. That's a silent mutation. You would never know by looking at the protein that there had been a mutation. Then there's a missense mutation, which is when it does change the message of the protein. So here now, we have a T instead of a C. So our mRNA has an A instead of a G. And so our codon is now AGC instead of GGC. So instead of glycine, we get serine. So we have replaced the amino acid. This is a missense mutation. So it still makes sense. We still have a protein, but it has a different message. So there's a couple of different things that can happen with this. Either that amino acid could make no difference. Maybe glycine and serine have very similar structures. Maybe they're both acidic amino acids. So there are groups. When we replaced one acidic R group with another acidic R group, it doesn't cause the protein to shape to fold up differently. So the protein's still going to be fine and still do its job just fine. Or maybe this amino acid is in a part of the protein that doesn't really have much to do with its job. Maybe it's at the end of a protein, it's nowhere near the active site. Maybe even if it's a significant change, it's not in a place that's affecting the function of the protein. So it's going to have little to no effect. Or it could have a very large effect. If that serine is now in the active site of the enzyme and the substrate can't bind anymore, that protein can't do its job. That enzyme is totally non-functional. Or if that serine is a totally different kind of amino acid than the glycine and the protein folds up entirely differently, then suddenly the protein can't do its job anymore. If its conformation, if its structure is completely different, it's not going to be able to do its job. So missense mutations can either be very, very devastating and very drastic, or they can have little to no effect. So there's a wide spectrum on missense mutations. Sickle cell is a missense mutation, right? Because there's one base pair substitution, and it brings in a different amino acid, and that different amino acid causes your hemoglobin to fold up differently. And because of that, it causes the entire blood cell to sickle. So just that one substitution led to a different amino acid, and by changing one amino acid, we totally changed the structure of the hemoglobin. But there's also several other missense mutations where it doesn't change the structure very much, doesn't have that big of a deal. So there's a big spectrum on missense mutations. Then we get to nonsense mutations. Nonsense mutations appear when there is a premature stop codon. So if you look up at the wild type, there's a stop codon at the end. We have UAA at the end. We made our protein, and we're done. However, if you look at this one, we substituted A instead of T from the wild type, and now we have a UAG instead of an AAG. So instead of bringing in a lysine, suddenly now we have a stop codon. So we got as far as the first amino acid, and now we're done. The ribosome reads it and says, okay, we're done. So our protein doesn't even get made. So our nonsense mutations are very, very devastating. They almost always result in a non-functional protein, and that's bad. If your proteins don't work, your body cannot really do anything. So missense mutations due to a premature stop codon are very, very devastating. So those are our substitution, or our single base pair substitutions. Then we have insertions and deletions, which, if you remember, mRNA is read 3 bases at a time. So CCC, GTA, AUG, whatever. There should be no T in there because it's mRNA. But if we throw one in there, now our reading frame is off. So instead of reading the correct 3, we shifted it. And that's going to cause a problem because everyone that comes after that is going to be affected. So these ones, our insertions and deletions, generally have a much, much bigger effect on the protein than do our single base pair substitutions. And these are called frame shift mutations because our reading frame, our 3 codons that we read at a time to bring in the amino acids, is off. Let's take a look at them. So again, here's our wild type. So this is an insertion or deletion that causes immediate nonsense, meaning it causes a premature stop codon, just like our nonsense mutations from before. So here we threw in an extra A. So we still have AAG, TTT, GGC. So it's the same as up here, except we threw in an extra A. And so the cell doesn't read AAG because it reads 3 at a time. So it doesn't know to not start right here. So it starts at UAA instead of simply reading AAG. And now UAA is a stop codon. So the protein doesn't get made. That's because of one base pair being thrown in there. So it has devastating effects. That causes immediate nonsense. Same thing would happen if we took out, there's no other examples in here, but if you took out one and you had a premature stop codon, that would be also immediate nonsense. Then sometimes we take out one and it causes extensive missense afterwards. So instead of one amino acid being off, all of them are. And that's really, really devastating. Think about the fact that what happened when we had one wrong with sickle cell. So think about all of them being wrong and the drastic effects it's going to have on the proteins. So here we took out one A, and so in our mRNA we have a U missing. And so what happens is our first two are right, but then suddenly, instead of reading UUU, we read UUG. Okay, so we have a leucine instead of a phenylalanine. And so if you look up here, the next one is GGC and then UAA, and they're still down here, GGC, UAA, except we're not reading starting here because we read 3 at a time. So now we're reading starting here. So GCU is a totally different amino acid than GGC. And following that, our stop codon's gone because the stop codon was right here, but we changed our reading frame, so now our stop codon is totally gone. Now we have AA with whatever follows. So everything that follows the amino acid that had the substitution or the insertion or deletion is going to be off. So just think about the drastic effects of that. If one amino acid can make such a difference as to cause sickle cell, think about what's going to happen here. It's very devastating. But then sometimes insertions or deletions can cause no problem whatsoever if they happen 3 base pairs at a time because of our reading frame. So here, for whatever reason, TTC got taken out. So down here, we were missing AAG. So we still have our MET, phenylalanine, and glycine, and our stop codon. So everything that follows is unaffected because our reading frame hasn't shifted. We are missing that one amino acid, and that could be really devastating if it alters the structure of the protein, or it might have no effect at all. If that lysine is near the end of the protein and it doesn't really have any function with anything, then it's not going to cause any problems. But if it is like in the active site or something, then it could cause some serious damage. So again, this is one of those ones that could have drastic effects or not so many. But inserting or deleting 3 nucleotides is going to have a far less drastic effect because there's no frame shift like there would be with 1 or 2 insertion or deletion. And so where do mutations come from? That's a good question. They come from things called mutagens. And so we can get mutations from our DNA machinery. So from DNA replication, from recombination, from DNA repair, from any number of things that your body's actually regulating with the DNA if it makes a mistake. But remember that our bodies are super good at regulating that. DNA polymerase proofreads, then we have mismatch repair, we have nucleotide excision repair, so our bodies are really good at regulating that. But then there's also external factors, such as chemical or physical agents that cause mutations. These are called mutagens, and these are things like sunlight, that's how humans get skin cancer, things like tobacco or other smokes or alcohol or anything like that. Those can cause really drastic mutations as well. And so our bodies are good at fixing them, but if you introduce your bodies to a lot of mutations, eventually they're going to build up and you're probably going to get cancer or get some drastic effect that you don't want. All right, 17.6 really quickly. Basically, there's 3 domains of life: bacteria, Eukarya, and Archaea. We are eukaryotic cells, part of Eukarya, and Archaea and bacteria are both prokaryotic cells. So bacteria and eukaryotic cells have a ton of different machinery. Our RNA polymerases are different, our termination of transcripts are different, our ribosomes are different, but Archaea tend to resemble Eukarya in these sorts of things. They tend to be similar to us. The cool thing about bacteria is that they don't have a nucleus, so their transcription and their translation is not separated. Ours is separated by a nuclear envelope. Transcription happens in the nucleus, translation happens in the cytoplasm, but that doesn't happen with bacteria. So they can do transcription and translation at the same time, which ups their ability to make a gene product, to make a protein. You can do it so fast because look at this. Here's their DNA molecule. As their DNA molecule is being turned into mRNA by these RNA polymerases that are right here, the ribosomes hop right on and they start making gene products. They start making polypeptides. So they are very, very efficient at making their proteins because of the fact that they're not separated. Archaea are likely do the same thing, but we don't know too much about them. But remember, humans do transcription in the nucleus and translation in the cytoplasm.